So as I just mentioned, from this point through the rest of the quarter, we're going to be somewhat off kilter of what the syllabus says because this lecture is ordinarily a full hour and 15, and now it's going to be 45 minutes. We'll just roll each lecture into the next one. So we're going to get as far as we can today, and then finish it up in the beginning of next class and get as far into metabolism as we can the next one. This lecture deals with protein trafficking. We talked about DNA being made to RNA, being made to protein, and now all of these other elaborations that could regulate either mRNA abundance or protein expression in cells. The last thing that we want to cover about how you go from the genome to cellular function is how do those translated proteins get to their respective locations inside the cell. And we'll be introducing that around this in the context of another syndrome called Noonan Syndrome, which is a congenital disorder, congenital meaning that a person is born with it. I have a couple of images of Noonan Syndrome patients on the right-hand side. The symptoms are macrocephaly. We heard about brachycephaly in the previous lecture. Macrocephaly is a big head, proportion to the body. Hypertelorism is eyes set far apart in the head. And then there are also some features with the neck webbing you can see in a couple of these pictures. The incidence of Noonan syndrome is 1 per couple of thousand live births. To give you some context on that, that's about the order of cystic fibrosis, another disease that we've covered in this class. The etiology of Noonan syndrome is very clear. It's due to an inherited defect in a signaling pathway called the Ras-MAPK kinase cascade that I'll speak just enough to provide context for this lecture, and then talk a lot more about it when we're discussing cell cycle and cancer, because it's an important pathway for cancer progression. Remarkably, these individuals that have a chronic defect in signaling through this cascade can survive to birth. And in fact, if their cardiac complications are handled, managed clinically, they can actually live to an old age. And it's really striking considering when you talk about this pathway and when you hear about what the role of this pathway is in coordinating cell cycle progression in a couple of weeks. Brief overview of the Ras-MAPK kinase signaling cascade, sheerly to introduce the relevant things you need for protein trafficking today and into next class. It's called a cascade because there's signaling information that's propagated from a high level on this pathway, and then the information, it's largely a phosphorylation cascade. So you have substrates that get converted to products, so they get phosphorylated, and then those substrate and products are themselves enzymes that, upon phosphorylation, catalyze additional phosphorylations inside the cell. The cascade overall is comprised of 3 types of proteins. First, which I want to speak more about in just a moment, is a so-called monomeric G protein. You've talked about heterotrimeric G proteins with Professor Barker. Do a compare and contrast of those in a moment. That leads to the activation of a serine-threonine kinase called RAF. It gets activated by phosphorylation, and then when active, phosphorylates a kinase called MEK. MEK is an interesting enzyme. It's what's called a dual specificity enzyme, a kinase that phosphorylates both serine-threonines and tyrosines. If we go back to amino acids, these are very different residues, and in fact what MEK is able to catalyze is a threonine adjacent to a tyrosine within two amino acids on its substrate called MAP kinase, which is another serine-threonine kinase that upon activation will phosphorylate a number of different substrates in the cell. Prominent among them are many transcription factors whose transcription factor activity is regulated positively by phosphorylation. So the net result of this cascade is upstream triggers from outside the cell activate Ras, RAF, MEK, ERK, phosphorylation of a whole bunch of transcription factors, change gene expression, change the regulatory state of the cell. And we'll give more details on this when we talk about some of those transcription factors and the impact of those post-translational modifications in a couple of weeks. But first I want to touch on Ras, because Ras has a particular localization in the cells, which bears directly on the material today, and it also serves as a good opportunity to revisit some of the material from earlier quarters. You heard, I think it was in quarter 1, about heterotrimeric G proteins. To recall, these are assemblages of 3 different proteins: G alpha, G beta, and G gamma. On the left-hand side here, and it's G alpha that is loaded up with GTP, and it's GTP loaded active state. It will hydrolyze that GTP into GTP, and that serves as that timing mechanism to shut it off. There are some recurring themes with the monomeric G proteins in that they bind GTP and they can hydrolyze. More to come on that in a couple of weeks. The key thing is there's no beta gamma, and the other key thing is that the similarity, that's a difference. One of the similarities, though, is that both monomeric G proteins and heterotrimeric G proteins are localized constitutively to the plasma membrane, the inner leaflet of the plasma membrane. And we'll talk about precisely how they are tethered covalently to the plasma membrane today into next class. Their localization and the defects in localization that are associated with Noonan syndrome tie everything together at the end. But to set the broader stage for protein trafficking, what I wanted to introduce in broad strokes is we did all the central dogma things, and even before that we talked about organelles, right? The subcellular constituents. How do different proteins know where to go inside of the cell? It's destined for the lysosome, or if it's destined to be secreted, or destined for the nucleus. After translation, how are those proteins routed or trafficked to their appropriate locations? And so this lecture has a couple of different facets to it that are logically connected by protein trafficking, but individually they're standalone units. To begin, what I wanted to introduce is the root of protein trafficking from outside the cell to the cell interior, and then we'll cover from the cell interior out to the outer portion of the cell, and then other things along the way. But first, on the way in. So if we have a protein that's, let's say, is on the plasma membrane surface that the cell wants to bring inside and localize to some other subcellular localization, this is the process of endocytosis. We've heard about this in a couple of different lectures: toxins getting endocytosed, other things on the plasma membrane surface. Upon endocytosis, these will go into an endosome that traffics into these endolysosomes or later lysosomes, where if it's something that got brought in from outside of the cell that is destined for hydrolytic degradation, those are broken up by those lysosomal hydrolases and things that were touched upon when we gave the original organelle pre-lecture in the beginning of class. Going from outside the cell to inside the cell is called a retrograde transport. So you're moving from things outside going in. The antonym of that is anterograde transport, which is, if you will, is the natural secretory and trafficking pathway to take translated proteins that are destined for either plasma membrane localization or secretion from their intracellular compartments. And more to come about the relationship between the rough endoplasmic reticulum, this thing called the Golgi apparatus that we've only mentioned by name, and then how these get to the plasma membrane in just a couple moments. First, endocytosis. In this cartoon, these membrane invaginations, okay, so these little cups here that form are the result of what's called a clathrin-coated pit. We'll talk more about clathrin in just a moment. Where does the pit come from? The pit comes from the original identification of these membrane invaginations from electron microscopy. They were just electron-dense holes in the membrane, and therefore they just called them pits. It wasn't until we developed freeze-fracture methods that gave a more precise molecular definition of what was happening at the plasma membrane as that endocytosis event was occurring. Now, what you have here is membrane down on the screen, and then one of these invaginations coming on the inside, and this is looking from the cytoplasmic side of the cell. This is clathrin, and it's the formation of what's called a clathrin-coated vesicle. Clathrin is a biomolecule. It's a protein. It has 3 heavy chains and 3 light chains that are shown schematically on the left here in red, and they form what are called triskelions. And a triskelion is, well, first let me a little bit more about clathrin. So there are sets of 3 heavy and 3 light. On the slide, it looks like they're just a flat triangle, but they're not a flat triangle. They have an angle associated with them when they assemble, like so, like a tripod almost. And they also self-assemble on the plasma membrane when you're forming a clathrin-coated vesicle. So you take these triangles and assemble them the way in which they're shown on the slide there. What you're going to get is a natural curvature if they're also interacting with the membrane. They're interacting with one another. They're interacting with the membrane, and by virtue of that tripod nature, you get this triskeleton forming around the membrane, and that's what gives rise to the curvature. Clathrin gets recruited to the plasma membrane through what are called adapter proteins, whose role is to connect the clathrin machinery, or clathrin heterohexamers, I guess you'll call them, to receptors on the plasma membrane surface. You have transmembrane receptors, plasma membrane, clathrin floating in the cytoplasm. When there's a recruitment of an adapter molecule to pull the clathrin to the receptor, that's the cellular trigger for endocytosis and internalization. Adapter proteins generally are any class of protein that bring 2 molecules together that ordinarily wouldn't interact. And as I think I already said, this idea of clathrin recruitment to transmembrane receptor, and oftentimes signals emanating from that receptor being the trigger for the adapter protein recruitment and the clathrin recruitment, is called receptor-mediated endocytosis. So there's some receptor event that's transmitted to the cell, and then that initiates the assembly of the clathrin-coated pits. Here's a cartoon of the early events of the process, and then we're going to lead into some of the later events. Let's say we have a transmembrane receptor here that binds to a ligand outside of the cell. And let's say in this example, that ligand is a nutrient. Maybe that's something that the cell wants to internalize and use in its metabolism. You have receptors on the plasma membrane surface for it. The ligand comes in, bind the receptors. Usually there's some type of signaling from the cytoplasmic domain of that transmembrane receptor. And by virtue of that signaling, that promotes recruitment of the adapters and recruitment of clathrin. Coated pit up here on the top. As those triskelions continue to assemble on the plasma membrane surface, you're going to get a more and more complete rounded pit on the plasma membrane. But clathrin itself can't finish the deal. All it can do is form that membrane invagination, and then it just hangs out there. What's required is a GTPase, so an enzyme that hydrolyzes GTP called dynamin. For some people, dynamin is a motor. We talked about motor proteins before. And so what's the motor activity of dynamin by virtue of its GTP hydrolysis? Dynamin is a twisting motor. This is why it's a little bit debatable if it's a real motor or not. But what it does is, you go to the apple orchards here and stuff. How are you supposed to pick the apples off the tree? You have the apple there, and you're supposed to twist it. Right? And so this twisting function is what dynamin does on that membrane invagination that was initiated by the clathrin-coated vesicles. So the clathrin gets the apple formed, and then Dynamin comes and picks it off. Once the vesicle has been completely internalized, there's an uncoating process where the clathrin and adapter proteins are recycled back into the cytoplasm for another round of endocytosis. And then there's a question of what happens to this vesicle. Here, there's a number of different paths. Different receptor-ligand interactions can go on different paths. There's also, for example, one receptor with two different ligands can go on two different paths. And similarly, one ligand with two different receptors can go on different paths. But I'll give you a sense of what are the routes that it could occur. First, in some scenarios, if you had this cell is not polarized at all. It's just a big balloon as far as this cartoon is concerned. But let's say you had an epithelial cell, apical side, basolateral side, a gut epithelial cell. Nutrients taken in from the lumen of the gut, internalized and endocytosed. Some of those may need to go right through those epithelial cells and get delivered into bloodstream or to neighboring cells. In that setting, you don't want to do anything to the ligand that got internalized. And so the process of transcytosis simply takes things on one side of a polarized cell and release it on the other side. So that can happen. Second thing that can happen is that as that, so now here you're an endosome. As that endosome matures, and what I mean by maturing, I mean fusing with other vesicles that are more lysosomal-like, including things that have pumps to pump in protons into the lumen of those vesicles. So as this endosome is maturing, it's getting progressively more and more lysosomal-like, and by that I mean more acidic. So you have this maturation of the endosome, and depending upon the nature of the receptor-ligand interaction, what can happen is as you change the pH, what was originally a strong binding event now can fall off. I think, where did we see this? How you acid strip an affinity chromatography column when we did protein purification. Right? So you change the pH, all those electrostatic interactions get disrupted, biomolecular interaction falls apart. So when that falls apart, for certain ligands that are not going to be fated to transcytosis, the ligand, if that was a nutrient that the cell wanted for itself, would continue to get degraded in the lysosome. They'll get chopped up into amino acids, fed into metabolism, which you haven't heard anything about yet, but brought into the cell, and now you have a nutrient source for the cell to take. If the receptor falls off from the ligand, there is an opportunity for that unbound receptor to get trafficked back to the plasma membrane surface through a process called recycling. So recycling is good for us, recycling is good for the cells, because they don't have to make that transmembrane receptor again. They can double use it. It's a lot of energy, as you've already seen, to translate a long polypeptide and traffic it to the plasma membrane surface. Therefore, if you can reuse them for multiple ligand binding events, that's going to be energy efficient. Conversely, if the ligand stays bound to the receptor, there are some interactions that are so tight, it doesn't matter if you're at endolysosomal pH, they'll still stay stuck to one another. Then what happens is that the ligand, if you will, drags the receptor down that lysosomal degradation route. This can be an important mechanism for receptor downregulation if you have a signal that's too strong from outside the cell. Let's run a hypothetical scenario where you have this is not a nutrient, but is a growth ligand that says proliferate, but not too much. Proliferate now, but then don't hyperproliferate. As the ligand comes in, the first round will send proliferative signals by ligand receptor engagement. But then over time, if that ligand really tightly binds to the receptor, and then the receptors get downregulated, the cell will eventually become numb to that proliferative stimulus and then will not continue further. So it's a mechanism for post-translational negative feedback, akin to some of these other negative feedbacks that we've talked about in other settings. How are we doing so far? Yeah? Okay. Well, I see the thing. Is there a question or? Okay. Yeah. That's why I have a blank slide here. Say pause, because that was a lot. On the one side, that's all you need to know about internalization and endocytosis, but that was a lot. Let's do the flip side now. Anterograde transport. How do you get those transmembrane receptors or secreted proteins like the ligand? The ligand had to be synthesized inside a cell at some point. How did it get out there to be bound by another cell in the extracellular space. Here we're going to talk about this upper pathway in greater molecular detail. And to do that, we need to categorize the different ways in which proteins can get imported into certain organelles, lumens or other vesicles inside the cell. There are two major categories. One is co-translational import, which refers to protein localization that occurs while the protein is being translated by the ribosome. When we did translation, we just had ribosome translation happening and no other things happening concurrently. That was a lie. All right, there are other things that are happening to that translated polypeptide and the mRNA that are important for proteins that need to get eventually secreted into the extracellular space, localized to cell membranes like transmembrane receptors, or as well as for proteins that need to go to the lumen of lysosomes, such as lysosomal hydrolases. We'll contrast co-translational import with another form of import called a post-translational import, which is two separate things. It means translate the protein in the cytoplasm, and then after the protein has been fully translated, relocalize it to another organelle. And this comes into play for nuclear localized proteins and mitochondrially localized proteins. Are these concepts limited to eukaryotes? Yes, because eukaryotes have organelles. For prokaryotes, there are other rules, for instance, dictating things that go to the cell wall versus into the protoplasm or things, but there's not anywhere near as much compartmentalization, especially by an organelle basis. There's absolutely molecular assemblies that form in prokaryotes, and so I don't want to get across the idea that bacteria are just a big soup of enzymes, but there aren't those subcellular organelles. That's one of the defining characteristics, prokaryotes versus eukaryotes. Okay. What's true for both of these is that the information about where a protein should go is encoded in the amino acid sequence. These are called signals or sequences or signal sequences. We'll get more details on that in a second. But the first point to get across is that that information is encoded in the amino acid that gets translated. So here are a couple of the rules of thumb related to protein trafficking in general. The first is that without any signal or targeting sequence to instruct extracellular, nuclear, mitochondrial, the default place for a protein to go is the cytoplasm. In many ways, when we did the translation lecture, that was what I was talking to you about, translation of a cytoplasmic protein. For co-translational import requires a amino acid sequence called a signal sequence. There are particular characteristics of it. I'll talk more about that in a moment. If a mRNA encodes for a signal sequence, the default localization of that protein will be to the cell membrane, the plasma membrane, or secreted outside of the cell. It can go to other places if they have additional sequences, and I'll speak more about that probably next time. So associate co-translational import with a signal sequence. And I put those two together, that's the term. It's unfortunately not very descriptive, but that's the term, is the signal sequence. Post-translational import, this is the thing for nuclear and mitochondrial, also have sequences, but they're called different things. But fortunately, they are more descriptive. Nuclear localized proteins have what's called a nuclear localization sequence, or NLS for short. And this is a sequence that will interact with other biomolecules in the cell that help recruit that protein into the nucleus. Similar themes, but I'll be giving less details on the mitochondrial targeting sequences, or MTSs, that similarly, if put on a protein, are necessary and sufficient to put that protein into the mitochondria. And I harped on that sufficiency aspect. If you took a protein that ordinarily isn't supposed to be in the mitochondria, for example, and you genetically, we did some genetic engineering, you use some genetic engineering, you put a mitochondrial targeting sequence on it, you have a good chance of getting it to the mitochondria. You put an NLS on it, good chance of enriching it in the nucleus. And then the last rule of thumb, and more, you'll have more understanding on this when we talk about it, is that proteins that are smaller than, there's some leeway on this threshold. If you're in the vicinity of 40 kilodaltons or smaller by size, and without any additional localization sequences, those proteins expressed in the cytoplasm will diffuse freely between the cytoplasm and the nucleus. The pores that act as the gatekeeper for the nucleus and the cytoplasm are too big to provide a sufficient restraint for small proteins. So small proteins will just go in and out. Question? That is a fascinating question. I've never had that asked before. And so your question is the post-translational versus co-translational distinction. Is that because of the number of membranes that need to be overcome to get that protein localized to one place or another? I don't think that's the explanation, but the thing that it just triggered my mind is I wonder if it may have to do with the evolutionary origins of how some of these organelles arose in the first place. So first one, to be clear, going from the cytoplasm to the nucleus does not require traversing a membrane. And you'll see more about that in a moment. Mitochondria, yes. But the reason why I'm still thinking about the answer to your question is that a lot of the thinking about how eukaryotes evolved is by this remarkable fusion of two different cell types, an archaea and a prokaryote. And the prokaryotes eventually, there was a mutual coevolution, and the prokaryotes eventually became our mitochondria. And so there's you have a situation of very early in evolutionary time, right, but late enough from the origin of life that cells were already probably localizing things to their plasma membrane, which is what might have been reflected in the co-translational import. And then over the comparatively later evolutionary time, you have to come up with these additional means to get proteins back and forth to these other organelles. That's speculation, though. We will begin with co-translational import, and this is going to be a zoomed-in version of that anterograde transport in the rough ER. This is zoomed in on the rough endoplasmic reticulum in that earlier slide. What are the sequence of events for co-translational import into the endoplasmic reticulum? And specifically the rough endoplasmic reticulum, I think this was said in the pre-lecture, the reason why the endoplasmic reticulum is rough is because it is decorated with ribosomes all on its surface, the membrane surface. And the reason why it's decorated with ribosomes, it's because those ribosomes are doing this. What are they doing? And how do they get there? They're not there all the time. What occurs is that if a ribosome is assembled, right, we're in eukaryotic world now, so this is 60S and 40S, right? Large and small subunits assembling on to an mRNA in the cytosol, okay? Begins the translation, start methionine, and off it goes, elongating the polypeptide. The signal sequence, the one that is the trigger for co-translational import, is on the N-terminus of these proteins. So it's an N-terminal signal sequence. I don't have to say N-terminal because all signal sequences are on the very N-terminus. When this signal sequence is translated by the ribosome, it is recognized by a protein called SRP. SRP stands for signal recognition particle, and signal recognition particle recognizes signal sequences. It doesn't seem like a circular argument, but that's what it does. And that ability to recognize that translated signal sequence, once that SRP has been recruited, then takes that complex and recruits it to the ER membrane. And the means by which that recruitment occurs is by an interaction between SRP and the SRP receptor, whose job is to bind to SRPs that have recognized signal sequences. It pulls SRP, the signal sequence, the ribosome, and the whole mRNA to the surface of the rough endoplasmic reticulum. And there's a particular assembly of proteins. I'll show this in more structural detail in a slide or two, called a Pore protein, which is what enables the translated protein to go from the cytoplasm to the lumen of the ER. And there'll be more details on that in a moment. There's some GTP binding here that I'm not going to emphasize too strongly. But what occurs by that GTP binding, hydrolysis, you translocate this translating polypeptide into the pore of this pore protein. So this is a channel that connects the cytoplasm to the lumen of the endoplasmic reticulum. And the nascent polypeptide gets threaded into that pore protein. The signal sequence is retained in the pore protein, as shown here in the green. As the ribosome is continuing to translate the rest of the polypeptide. It's threading the translated polypeptide through the pore protein into the lumen of the ER. Translation, translation, translation. When it encounters a stop codon, release factor, disassembly of the ribosome, polypeptide is all the way inside, and then upon completion of translation and disassembly of the ribosome, there's a signal peptidase that's in the membrane of the ER that becomes activated, cuts off the signal sequence, and now you have a mature translated polypeptide in the lumen of the endoplasmic reticulum. And then probably this is the best point to remind that the interior of the endoplasmic reticulum and the interior of all vesicles, lysosomes, endosomes, Golgi apparatus, which we'll talk more about in a bit, those are all topologically identical to the outside of the cell. So if this and nothing more, this protein will be destined for secretion. Another thing to say is that even though in translation, for example, I emphasize that all translated proteins start with methionine or formal methionine, but here for methionine. This is true, and this is true for the proproteins of secreted proteins destined for secretion. But note that that methionine is in the signal sequence that got chopped off. So mature secreted proteins don't always start with a methionine. Their precursor protein did, but the mature one that gets secreted doesn't have to. Here's what that assembly of ribosomes, this movie doesn't work, signal sequence, and then the pore protein shown here looks like. This is the pore protein. This is so-called Sex61 complex. It's a heterotrimer. You can see the different colors here, and there's some flexibility to this pore protein, and more to come on that in a moment, because you need some of this flexibility to show the next thing that the pore protein can do. Yes? Yeah, is the signal sequence a conserved sequence? It is. I'm sparing you what the consensus sequence is, but it is. You can look at a translated protein and bioinformatically say this is a signal sequence. So there are characteristics of that signal sequence that are recognized by SRP, and this is how you could look at a new protein in eukaryotes and say this looks like it should be secreted. And those were defined by the same tools that I introduced when we discussed consensus sequences for origin or replication. Same idea, but you're doing it with amino acids rather than nucleic acids.
You might have talked about this earlier, but when you have co-translational reporting of protein, it takes a lot from it.
Yes. Yes. I'm going to reiterate it for the video. With co-translational import, the translation starts in the cytosol, and then once the signal sequence has been translated, SRP and that SRP receptor interaction pulls the whole macromolecular complex, mRNA, our ribosome, nascent polypeptide, down to the pore protein. And so that's the way that secreted proteins are made. What about transmembrane proteins? That won't appear to address the transmembrane protein issue. Here we have another type of sequence called either a stop transfer sequence or a topogenic sequence. What the topogenic just means is it provides information on the orientation and the topology of a translated protein in the membrane. The characteristics, we were talking about amino acid sequences and what are the things that are characteristic for the signal sequence. Topogenic sequences are straightforward. 20 hydrophobic amino acids, just like that hydropathy plots that you did Q1, so that everything comes back. All right? That's the criteria for a topogenic sequence. Envision you have, I don't like this cartoon, but I've never gotten the wherewithal to fix it, so I'm just going to tell you what all the problems are with it. All right, so for this cartoon on the left-hand side, there's supposed to be a ribosome here. All right, so the ribosome is translating. You don't necessarily need, we're going to talk about this for this particular category of transmembrane proteins that also have a signal sequence. You don't need to have a signal sequence to become a transmembrane protein, but it started here with the pore protein in the ER membrane. Ribosome is translating. And then now let's say it comes across one of those topogenic sequences, stop transfer sequences, hydrophobic. It gets translated, threaded in through the pore, but it's hydrophobic. So there's not going to be any thermodynamic driving force for that stop transfer sequence to want to continue to get threaded in because the lumen of the ER is just as hydrophilic as the cytoplasm. What happens is it gets stuck. So it gets stuck in the interior of the pore. And because of the, and again, I can't do more than just hand wave on this, the flexibility of that heterotrimeric complex that forms the pore protein, they can wiggle relative to one another. And if you have another hydrophobic sequence in the middle, bear with me with some wiggling, that hydrophobic sequence can wiggle its way through, and now it's in the ER membrane, along with the phospholipids and things that are in there. So this is how you get from inside the pore complex to in the ER membrane. The ribosome keeps translating, but now it's undocked from SRP and the SRP receptor and things. It just translates the rest of that protein out in the cytoplasm that is just adjacent to the ER membrane. And then the last thing I hate is don't get the impression that the signal peptidase just remains there forever embedded beyond. So that should have been removed. But this is how you'll get what's called a type 1 transmembrane protein by having a signal sequence on the front end, translation, first stop transfer sequence, and then translated out from there. This can happen again and again and again. G-protein coupled receptors, 7-pass transmembrane receptors. How the heck does that happen? That happens with 7 stop transfer sequences. So they encounter a hydrophobic sequence, wedged in the membrane, undocks for a little while, then it encounters another stop transfer sequence, back into the pore complex, back into the membrane, off again, on again, off again. All right. Give me one more slide, because then after this I go on a long soliloquy about sugar modifications. I want to talk in broad terms about additional modifications that happen on polyproteins in the rough endoplasmic reticulum and thereafter, after they've made it into the ER lumen. I'll redefine post-translational modifications. These are chemical changes that occur to a protein sequence after translation has occurred. We've seen these before. Cleavage, phosphorylation, methylation. These are all post-translational modifications. There are more. One of the ones that's really important and tightly associated with the endoplasmic reticulum is the formation of disulfide bonds. Remember the environments that are oxidizing and reducing inside the cell and outside the cell. Cytoplasm is a reducing environment. the lumen of all these vesicles in the outside of the cell are oxidizing. Cysteines that are ordinarily free thiols in the cytoplasm, after being translated and localized to the ER lumen, are now in an oxidizing environment, and therefore will be inclined to form disulfide bonds. Talked about these in the beginning of the class. In the ER, there's a dedicated enzyme called the protein disulfide isomerase that explores different disulfide bond configurations to arrive at the most thermodynamically favorable one. Occasionally, it's the one that's in closest proximity, but in the case that it's not in a thermodynamically favorable conformation, it will reduce one disulfide bond and then form another one and see how that improves with the thermodynamics of the folding. I scooped myself for item 2. This is where a proper folding of secreted proteins occurs, is in the endoplasmic reticulum. And in fact, if cells are making too much protein, or they're put in an environment like a high temperature, higher than the temperature in which they've ordinarily involved, that will cause protein misfolding. And if you have extensive protein misfolding, you'll get a backup of misfolded proteins inside the cell that triggers a whole regulated response inside the cell called the unfolded protein response that stops translation, sends off a whole bunch of stress signals, and if it lasts for too long, will commit the cell to undergo apoptosis, because it's a sort of a bad state of affairs. So all that quality control is occurring in the ER. This is also where, in addition to the disulfide bond formation, you can get proteolytic cleavages beyond simply the signal sequence getting cleaved off. Examples that you've seen. We had one slide way back when on insulin and the chains and everything. So insulin is synthesized as a protein that gets cut multiple different places and then reassembled. Those cleavages occur in the ER. That was one protein assembling, but multimeric proteins can also assemble. Think of antibodies, heavy chain, light chain, 2 different gene loci. Okay, how do they come together in that Y that you've seen many times before? How do they come together in just that right disulfide bond configuration? Endoplasmic reticulum. And then the last step that occurs in the endoplasmic reticulum, where it starts and then much more extensively in the Golgi, which is the next location for these proteins, is the addition and elaboration of sugars, carbohydrates, to particular residues on that protein. And this process of sugar addition and elaboration on proteins is called glycosylation. I think I mentioned glycosylation a little bit before. When class starts up after the holiday, we'll do a deep dive on protein glycosylation and then circle back to Noonan syndrome. In the minute that we have, are there other questions on co-translational import, protein trafficking? All right, have a nice holiday, guys.